One of the functions of the cell membrane is that not only does it keep things out, but it decides what's allowed to come in. It's selectively permeable. Now, sometimes it allows things to come in through protein channels, right? So we have channels inside, say, neurons that allow sodium to just rush right in. So there will be a particular ion channel specific to sodium, and it'll allow it in when the time is correct. But sometimes materials have to be brought in via bulk transport. And in that situation, it's definitely active transport because it takes a lot of energy to do this. You might have a food particle that comes up against the side, and the cell is going to essentially swallow this food particle. Could you go over the steps of meiosis and mitosis and the steps they go through after the terminal? Yes, I can. Yes, and you know what? These ideas are kind of related to each other. So the great thing about liquid membranes is that they can move. You can change their shape, and they can end up pinching together. So this little food particle, it's going to move its way in, and we're going to start folding this membrane into a pocket. Now, that pocket, when the two sides of this membrane here, right, they're going to come closer together. We're going to get a situation where the two ends are going to come closer together as we pinch this part together. So what's going to happen is we will get the food particle brought inside of its own little transport package, and the membrane will seal itself up. Right? Because it's a liquid membrane. It doesn't matter if you break a piece off. It's not permanently fractured. It's not like when you break a bone and then that bone is broken. It's liquid. You can't cut a liquid. You can't like take a butcher's knife and go right through water and cut it. As soon as it comes back together, it's healed. Right? That's the great thing about having fluid membranes. Inside the cell now, depending on the size, what we have is a food vacuole. Vacuoles are a little bit larger than vesicles, and they're for storage. So we're just going to store this little bit of food inside. Now, when the cell is doing this, it's endocytosis, because we're taking material into the cell. Endo means into. Endocytosis can be broken up into different categories. When we're bringing in solids, like we did here, we call that phagocytosis. Phage means eater. It means to eat. So this is literally cell eating. We have a certain kind of virus that you hear about that attacks bacteria. It's called a bacteriophage, a bacterio eater. If we are bringing liquids inside, so phagocytosis, solids, pinocytosis, like a nice glass of Pinot Noir, liquids, bringing liquid from the extracellular environment into the cell. And so maybe you just want to bring in some water, or maybe there's some potassium outside of the cell and it could use it inside. It's going to bring in those liquids through pinocytosis, literally cell drinking. If we move in reverse, we have exocytosis, and that's mostly for excreting waste. But there's other things that the cell might want to excrete. So when you have two nerve cells that are synapsing, right? So I have here's a nerve cell body. It's got its dendrite coming out on the top here. Then you got these long axons, and you got more of these little processes coming off. When one nerve cell synapses with another one, they have to be able to exchange chemicals across these synapses. That's how they communicate with each other. And the way that they exchange chemicals is that one of them is going to do exocytosis to send those neurotransmitters out. So we're going to go from this neuron, and they'll hit protein receptors on the outside of the postsynaptic neuron here. So there's another reason why you might want to do exocytosis, is to communicate with other cells. But exocytosis is just the same thing in reverse. It's just moving material out of the cell. One more kind of endocytosis that we can talk about is receptor mediated endocytosis. This is where we have certain proteins, certain protein molecules that are called receptors, and they are floating around on this cell membrane here. Now, we saw the channels earlier, right? And the channels help with like facilitated diffusion. They'll just allow things to fall passively down their gradient a little bit faster than they otherwise would. But receptors are a little bit different. Receptors don't allow material in. They just detect the presence of certain molecules. So this is a receptor. It's kind of like it's kind of like a doorbell, actually, now that I think about it. This is a molecule that this protein is something that a molecule might bump into. And when it bumps into it, it communicates into the cell that there's something out here that might be interesting. The receptor's looking for one particular kind of molecule. Its whole life is just waiting for the right molecule to bump into it. But as soon as it does, it triggers endocytosis to occur. So that's what we call receptor-mediated endocytosis. And we saw an example of that when we were talking about genetics with people that have hypercholesterolemia. They don't make the receptors on their cell surfaces that detect the presence of these lipoproteins, of low-density lipoproteins. So because they don't detect them, they don't pull those cholesterol-carrying molecules out of the bloodstream, and they end up with much higher blood cholesterol levels than they otherwise would, because they fail to make the receptor that triggers endocytosis for cholesterol-carrying lipoproteins. So that's a good example of that there. But the reason why I say it's like a doorbell is because the receptor itself doesn't allow the material in. The receptor just communicates to the cell that it should do endocytosis to fold around this molecule. Even if there's just little trace amounts, it triggers it to do it. So when you ring a doorbell, right, the doorbell doesn't let you in, the door next to it allows it in. So that's where you would get receptor-mediated endocytosis.